Okay, so now moving on to some accessory organs, we're going to talk about the liver and the pancreas and the gallbladder. So these are accessory organs that are associated with the small intestine. If we're talking about the liver, the liver has tons of metabolic and regulatory roles, but we're really just going to focus on the digestive function of the liver for this chapter. And its digestive function is really for producing bile that's going to be exported to the duodenum, or that first part of the small intestine. What is the function of bile? Bile is a fat emulsifier, meaning that it's going to help break up fats into smaller particles to make them easier to digest. Our gallbladder, its only function really is to store the bile that's being produced from the liver itself. And then if we're talking about the pancreas is going to be the organ that's responsible for producing most of the enzymes that the digestive chyme actually needs, as well as some bicarbonate ions that are going to help neutralize the stomach acidic secretions. So again, a lot of this section, or a lot of this chapter even, when we're talking about the anatomy of the liver, most of it is going to pertain to lab, and you won't be tested on it for lecture. I'm not really super concerned with a lot of the arteries and veins. You will have to identify those in lab, but for lecture, just kind of know the major ones as presented to you in this PowerPoint. So you probably learned about the greater and lesser omentum. Well, it's the lesser omentum that anchors the liver to the stomach itself. We have the hepatic artery and vein. Where do these enter? They enter through the porta hepatis, which is, let me use the highlighter, right here. We also have bile ducts that are associated with the liver, as we would expect, because our liver is producing the bile. So the common hepatic duct is going to be the one that's leaving the liver. We have our cystic duct, which is going to connect the liver to the gallbladder. And then we have our bile duct, which is formed from the union of the common hepatic and the cystic ducts. The cells that make up the liver we call hepatocytes. One of the unique characteristics of the hepatocyte is that they have a larger amount of both rough and smooth endoplasmic reticulum, as well as Golgi apparatus, peroxisomes, and mitochondria. If we think about what the function of these organelles are and what the function of our liver is, this makes sense. So if we're talking about the functions of the hepatocytes, well, one function is going to be to produce the bile, around 900 milliliters of bile. We're also going to be processing blood-borne nutrients. So for example, one of the nutrients is sugar in the form of glucose. Typically, we store glucose as a polymer, and that polymer is glycogen. We also need to make huge amounts of proteins, plasma proteins. So we would expect to see lots of ribosomes or rough ER, as well as the Golgi apparatus, which is responsible for tagging the proteins and knowing where to send them. Store fat-soluble vitamins, and detoxification is huge. So smooth endoplasmic reticulum, or the smooth ER, its primary role is in detoxification and lipid production. Okay, so let's talk a little bit more about bile. What is bile? Bile is really just a yellow-green alkaline solution. And like I told you before, its goal is to help break up lipids and kind of make them into more manageable size particles. So we have bile salts that are in it. These are derived from cholesterol, and they help with breaking up the fat, and they also help with absorption. Bilirubin, this is a pigment, and if you remember back to our blood chapter, the waste product, bilirubin is a waste product of heme from red blood cell breakdown. Bacteria break down in the small intestine, and they turn bilirubin into stercobilin, and that's what gives our feces the brown color. We also have cholesterol, triglycerides, phospholipids, and electrolytes in there as well. Only bile salts and phospholipids are actually aiding in the digestive process. So, enterohepatic circulation, it's a recycling mechanism. It's really important because it's going to help conserve the bile salts that we are using to help break these things down. So, when we're talking about the enterohepatic circulation, we're talking about reabsorbing these bile salts into the blood by the ileum and returning them to the liver via the hepatic portal vein or blood. So we're going to resecrete them into the newly formed bile. So out of all the bile salts that we're using to help break up the lipids, 95% of them are recycled back to the liver to be reused. Because we're using huge amounts of bile salts, anytime we can recycle something, that's great. We're going to take that route because it's going to require less energy because we don't have to resynthesize as many. So this is that process that we were just talking about of recycling our bile salts. So here's our bile salts being produced in the duodenum, or secreted into the duodenum right here. The bile salts are going to travel through the small intestine and help with lipid digestion by breaking them up into smaller pieces and also help with absorption. And then as they travel through, 95% of the bile salts are going to be reabsorbed by the last segment of the small intestine, which is called the ileum. And once we reabsorb them, we're going to recycle them back via the hepatic portal vein back into the liver. So we only have to synthesize about 5% of bile salts or new bile salts to replace the correct amount. So just talking a little bit about some clinical balance, gallstones, I'm sure you've heard of. So we're talking about bile being the major vehicle for excreting cholesterol from the body. And bile salts keep the cholesterol dissolved within the bile. So too much cholesterol or too little bile salts allows for cholesterol to crystallize. When cholesterol crystallizes, it forms gallstones. And once we get these large crystallized cholesterol, they can actually obstruct the flow of bile from the gallbladder. When the gallbladder or its duct contracts, these really sharp crystals can cause huge amounts of pain. And basically, how do you diagnose if you have gallstones? You have to go in and do some ultrasound imaging. As far as treatment goes, there's different sources of treatments. Some can just be dissolve those crystals using drugs. Other ones have to use vibrations from ultrasounds. Now they're using lasers to help vaporize them, or you can go in and actually surgically remove the gallbladder. Okay, the pancreas. The pancreas is really important in the digestive system because it produces the enzymes that break down all macromolecules that we are bringing in. So all macromolecules, we're talking about proteins, lipids, and carbohydrates. So the enzymes secreted from the pancreas can break down all of these. Before, when we're talking about saliva in our mouth, we're focused on salivary amylase, can only break down carbohydrates. We're talking about pepsin secretion in our stomach that could only break down proteins. We had a little bit of lipase breakdown from lipases, enzymes that can break lipids down. But our pancreas is secreting all the enzymes needed to break down all of the macromolecules that we need them to break down. That's its predominant function. So the pancreas contains both exocrine and endocrine functions. We're talking about the exocrine part of the pancreas. We're talking about the production of pancreatic juice. So we have acini, and these are clusters of secretory cells, and they produce zymogen granules that contain proenzymes. So proenzymes are things that not are in their active form yet. They need something to come in and activate them. And then we have ducts, and the duct cells are going to produce water and bicarbonate or alkaline secretions. The endocrine function of the pancreas is secretion of insulin and glucagon, what we learned about in the previous chapter. And those two hormones are going to be secreted by the pancreatic islet cells. So composition of the pancreatic juice. We produce about 1,200 to 1,500 milliliters of pancreatic juice every day. It's predominantly water, but we also have some enzymes and electrolytes in there. When we're talking about the electrolytes, it's predominantly those bicarbonate ions. The pancreatic juice is also has a high pH, so a pH of 8. So that means that it's actually basic. And the purpose of having this basic pH of the fluid is to help neutralize the acidic chyme that's coming in to the duodenum itself. Also, we need this elevated pH in order for our intestinal and pancreatic enzymes to perform optimally. So important enzymes, proteases. If we're talking about a protease that's going to digest proteins, like I mentioned before, it's secreted in the inactive form, just like when we talked about pepsin in our stomach. Anytime you see an amylase, that's going to be for carbohydrates. Lipases break down lipids, and nucleases break down nucleic acids. So proteases are secreted in the inactive form, like we talked about before. They are activated after they reach the duodenum itself. So we have enteropeptidase, which is an enzyme bound to the plasma membrane of the duodenal epithelial cells. It activates the pancreatic protease, which is trypsinogen, into trypsin. So now we've talked about two different proteases or protein enzymes that can break down proteins. We talked about pepsinogen being activated into pepsin by hydrochloric acid in our stomach. Now we're talking about a different protease or enzyme that can break down protein. And this one is going into our duodenum, but it's being produced by our pancreas. And we're talking about trypsinogen, which is the inactive form, which is going to be activated into trypsin. Once we have trypsin activated, it can activate more trypsinogen. And then we can also have pro-carboxypeptidase activated into carboxypeptidase and chymotrypsinogen activated into chymotrypsin. So basically, once we have activated trypsin or activated trypsinogen in the form of trypsin, trypsin can go on and activate other enzymes that break down proteins. That's the big picture there. So how do we regulate the secretions from the pancreas and regulate the secretion of bile into the small intestines? Well, bile and pancreatic juice secretions are stimulated again by both neural and hormonal controls. If we're talking about hormones, we're talking about cholecystokinin or CCK, as well as secretin. These include our 2-angastrones. If we're talking about, well, I guess we'll get to that later. If we're talking about bile secretion, that's going to be increased when our enterohepatic circulation returns large amounts of bile salts. If we're returning large amounts of bile salts, that means we are using large amounts of bile salts. So we're going to have to increase the bile secretion. Secretin also acts to increase bile secretion. So from secretin from the intestinal cells that are being exposed to hydrochloric acid and the fatty chyme, that's also going to be a trigger to stimulate our gallbladder to release bile. So again, what you're focused on here is what is the role of CCK and what is the role of secretin in both pancreatic juice and bile secretion. So I would kind of walk through this flow chart to try to understand what these two hormones do. CCK and secretin are going to be secreted by our duodenal enteroendocrine cells. If we're talking about CCK's function, CCK release is going to be stimulated by the presence of proteins and fats in the chyme. Secretin is going to be released and stimulated by the acidity of the chyme. They both enter into circulation. If we're talking about pancreatic secretions, CCK is going to induce the secretion by the acinar cells of the enzyme-rich pancreatic juice. Secretin acts a little bit differently. It causes secretion of the duct cells that bicarbonate ion. Bile secretion by the liver, bile salts returning from the enterohepatic circulation. Those are going to be your main source for or stimulant for bile secretion. Secretin doesn't really play a role when we're talking about a stimulus for bile secretion. As far as gallbladder contraction goes, CCK causes gallbladder contraction and would cause bile secretion because of the contraction. And if we're talking about hepatopancreatic sphincter relaxation, the CCK causes a hepatopancreatic sphincter to relax, and the bile and the pancreatic juice can enter into the duodenum.